genes directly. And so what have been some first with polychain chromosomes? That gene activity requires losing of chromatin. We now know that the nucleosomes are reopened up and so on. But this was something when puffing was discovered in 1930s and 40s and early 50s. Biermann explained that puffing is chromatin loosens and that shows chromosome opening. And this happened before we knew that genes are made up of DNA. This phenomenon was predicted long back, that there's a fiber which opens up and that becomes active. And of course, physical map, which in humans have been very important, but in case of Drosophila, physical maps were made as early as 1930s. Another important point that polychain chromosomes and Drosophila studies showed was the steroid hormones act on genes and induce cascading pattern gene activity. All the steroids that we talk about in humans, be it estrogen, progesterone, testosterone, that they act on directly on genes was discovered by directly exposing salivary glands to ecdysone, and then see that you see a cascade of gene activity. I'll show that later on. And then the whole process of heat shock induced gene activity was discovered in Drosophila because here you could see actual puffing change. I'll show this again later on. Then when a gene becomes active, now you have possibility of looking at that when a gene becomes active, how does chromatin change, what kind of molecules are associated there, what bring about what regulatory molecules, transcription factors, and bind there, and how the transcription machinery is activated. All this can be seen with these chromosomes. For example, physical mapping. See, because the homologs are paired, so if you have a small duplication, theoretically we draw it like, okay, deletion, that gene 3 and 4 are missing on this homolog and they are present in this. And we can see in polychain chromosomes, we concentrate on this point, that you have these bands which are missing on this homolog. And so immediately you can make out. Now, using this kind of technology, we can even map a promoter, as we had done way back in 1996, that we mapped a promoter by using small deletions at chromosome level, that when a particular region was deleted, the gene could not be activated. And so polychain chromosomes have provided enormous information. And I think looking at this under a microscope can really mesmerize students. They can enjoy looking at genes. And this is what we can see. For example, if you take late Thuricella larvae of definite age and you prepare salivary glands from them every 1 hour after, you can see the dramatic changes in puffing pattern. This study, which was done by Michael Ashburn in the 1960s, really established that, for example, this is pre-ecdysone. This particular first panel, A, is before ecdysone is given, and then as the larva is releasing ecdysone, or if you do it in vitro, you expose the salivary gland to ecdysone, and then withdraw ecdysone, and then you can see every hour these changes. For example, this was condensed here, condensed, it opens up much more, and then goes back, regresses. So we can see the dramatic change in gene activity, which we cannot see in any other cell because we can't see the genes when they are active in that. But polychain chromosomes provide that, and that's the advantage that I think we should take. Heat shock response was again an accident in from a normally grown larva polychain chromosome. This was on a heat shock larva. Now, if we concentrate on these 87C here, completely open, but 37 degrees centigrade for 5 minutes, or take a larva and expose larva to 5-10 minutes and make chromosome progression, we can see the changes, the new puffs. And that's what they're called as heat shock puffs. Later on, of course, they could be shown that associated with these heat shock puffs are synthesis of these new heat shock proteins. And so this opened up a whole field of stress biology, starting with these polychain chromosomes. And this can be done in any classroom. Only thing is, you will have to ensure that your control larvae remain at 24. If they're exposed to 30 degree or 32 degree, already heat shock puffs will become active. So that's one limitation here, that you need a controlled temperature room. The heat shock response can also be seen now today thanks to transgenic lines. For example, this particular line that we have also used and has been made long back makes use of the HSP70 genes promoter fused to beta-glucosidase gene, lag gene of E. coli. So now what happens is when these larvae, this is the gut of Drosophila larva, these are the big tubules, when they're exposed to elevated temperature, the HO70 promoter drives beta-galactosidase of E. coli, the lactogene of E. coli, and so beta-galactosidase is synthesized, which can be detected by histochemical staining using XCal as the substrate, which is yellow colored powder, but when acted upon by beta-galactosidase, turns into magenta blue. And so the whole tissue, every tissue of Drosophila expresses heat shock genes, and this can be seen very conveniently. The other area which Drosophila has really revolutionized is developmental biology. And this is very interesting thing. To begin with, Drosophila embryology looked very bizarre, completely different from what we have learned about cleavage divisions in frog or Amphioxus or mouse or sea urchin. Here, the initial zygote keeps on dividing nucleus without cell division. So you get a syncytial mass of embryo with thousands of nuclei and common cytoplasm. And then at a particular stage, which is equivalent to blastoderm stage, these nuclei migrate to periphery. Mind it, this is still a single cell. And these are independent nuclei aligned at the periphery. And then now the cells form. So the cleavage is, the actual cell division delayed much more. After about 13, 14 cycles of cell division, when you have thousands of nuclei in a common cytoplasm, you get this. And then there's no typical gastrulation. There's something called, equivalent to that, is germ bud extension. So this was very unusual. But now what we know is this unusual development has become a real advantage in learning how genes determine development. How genes determine that this should be head region, this should be thorax region, this should be abdominal region. This is what was done by using simple genetic tricks. And so this has been very simple way. And now we can look at these processes, but there are simple methods to look at these in any classroom, that how this development happens. For example, this is a simple thing which has been created by Dr. Shamla in the Mysore lab. They teach it, that you can collect eggs over a period of time in this petri dish with agar, using this kind of a bottle. You can make it at home and have these flies lay eggs on this. This is, for example, the petri dish containing these numerous white dots which are the eggs. And you can make a egg filtration device like this. Take any plastic bottle with a screw cap, remove the central part of the cap, but so that the rim can be still tightened, and put any fine cloth under this, nylon cloth or cotton cloth, clean, and tighten it so that the cloth will hold the eggs on its mesh. And now you can treat each of these eggs, pass them through different solutions without worry about losing. All that we need to do is, the egg is covered with a chorion layer. For example, these are the two chorion filaments, and the whole egg is covered with chorion layer. So that chorion layer, which is an opaque white layer, has to be removed, which can be done with simple bleaching powder. All you need is a 5% solution of bleaching powder. Treat it for a few minutes, and the chorion dissolves, wash away the bleaching, and now we have embryos which you can examine under even bright field microscope. Phase contrast is much easier, but even if you look in bright field, you can see these kinds of developmental events happening. The other thing that one can do is, if one has a fluorescence microscope, one can stain the nuclei in this with DAPI fluorescence stain, which can get into live cells, and then you can see the mitotic divisions and the nuclei very clearly all through. And this kind of experiments can be done in a classroom where students can see how the development happens. And what one can also notice is, at the posterior end here, one can see the first cells that are formed are the pole cells, and they are the exclusive precursors of germ cells. So one can see the difference between soma and germline if one does this kind of experiments. The other thing that we can look in developmental biology into Drosophila is how the adult organs are formed, adult structure, legs, wings, and so on, which larva doesn't have. And what we know is that within the body of the larva, we have these imaginary discs. These are hand drawings. These are the real discs that one can see in the microscope. They are located around the brain region. This is the larval brain, the brain ganglia, and this is the ventral nerve cord. And these are the different discs. Each one of them will give rise to an external structure. And for example, this particular we call wing disc, which has a shape like this. This part will become the wing itself. This will form the junction with thorax. This is the halter disc or the leg disc. This will form the antenna part. This will form the eye part, and so we can look at this. If we take very early viewpoint, dissect them, we can see the. For example, this is a developing leg, so segments are now forming. To begin it was like this, but then as it develops, it begins to make these segments. So one can do lots of experiments with these. Lots of genetic studies have been done to find out how does a cell know what it has to form and so on and so forth. But the other thing is that morphologically one can do these experiments in a very simple way. The other thing that Drosophila allows is the mutation, genetic mutation. E.B. Lewis, for example, who had worked all of his life on this bithorax phenotype. That means normally we have only one pair of wings. That's why we call them a diptera insects. The second pair is present as a very small halter. Because of this mutation in the bithorax gene, the second pair also develops as a full leg. And that's what Lewis examined, that what are the genes, what interactions, and he discovered these homeotic genes, and that's what led to the understanding of the Hox, homeobox, homeotic gene, that these genes, it's not, in Drosophila, for example, we know it from bithorax, but in humans also, same gene is present. Actually, the human gene will do same function in Drosophila. If you put Drosophila gene in mouse, it will do the same function, although they give us different structures. Because what we now know is what these genes are telling the cell is its coordinates in the body, whether it's in proximal part, distal part, and thereby regulating downstream genes. And this has been a remarkable discovery. Another mutation like this that we call homeotic mutation, which are discovered way back in 1949, is that you have this antenna, which because of the antenna PG mutation, antenna develops like first leg. Obviously, this fly will be a great research model because it doesn't have the antenna, but for geneticists, for molecular biologists, it becomes a very important way to understand what makes an organ develop into what it is. And one can look at this. And this is what the Nussbaum Pollard and colleagues, Eric Wishaus, again did a very simple genetic study, induced mutations in wild type Drosophila, look at embryo that die as embryo, and then examine why did they die. And they discovered these homeotic genes, the gap gene, the pair rule genes, and the segment polarity genes, and which even today we can see. This is very simple. You don't need any sophisticated equipment. All you need to do is look at the larva or embryo under a microscope, and you can see these segments, the identical patterns. If you're dark field, these images are dark field microscope, because then the identical patterns, which are highly intense, appear bright. And so one can easily make out that, okay, there is something missing here. That means the gene that are mutated are responsible for making this region. These were part of a gap gene mutation. And so you can have the different mutants and examine them. And of course, E.B. Lewis, Eric Wishaus, and Nussbaum Pollard got the Nobel Prize in 1994. And actually, I wrote a commentary on this, 1995, that this is a tribute to power of genetics. These Nobel Prizes didn't employ any molecular biology, any sophisticated equipment, simple plain genetic mutations and examination of the eggs, examine the adult flies, and how do they behave. And that's what led to their Nobel Prize winning discovery. And the important point is that any classroom can look at these phenotypes in a simple way with basic microscope and basic stereo binocular things. Mutations, we can see, we can cause a misexpression of a given gene, and then we can see that instead of this naked cuticle here between the segments, one can get these forming everywhere. So one can understand how the genes are regulated, how do they direct the cells to make what they do. We can also look at, again, using beta-glucoside reporter gene, lag gene constructs, where you use different promoters and put beta-glucoside downstream of it, make a transgenic fly, and then examine where beta-glucoside is expressed, so that without doing molecular biology, we can exactly know where the gene is expressing. And some of these can be done in classroom without any great difficulty. The other experiment that we can do is that a glycerol controls preparation. A simple experiment. You take a larva, take a fine thread, tie a knot around somewhere, and then see the larva doesn't pupate. Why? Because as you have tightened it, the ecdysone, which is secreted from processor gland located above brain region, doesn't move to the body, and therefore these cannot undergo uh, molting. And so this is again a simple experiment, and we can do it in the soft lab. No fear of animal ethics or cruelty on animals and so on. Fortunately, we can do molecular biology. We can extract DNA from flies. We can look at the DNA in a standard way, that the same protocol as we do for mouse or anything else. All you need is a centrifuge, and that's it, and flies. And then agarogel, so they can see the DNA, the marker, and the intact genomic DNA, shared DNA, or you can digest and can see different fragments. So these are again Drosophila flies you can get in large number. Now there is a protocol that we can isolate DNA from a single fly and examine it. And that's where the sensitivity has become very useful, that a lot of molecular studies can be done with this kind of technique. We can look at if there's any environmental toxicity, xenobiotics are there or something else is damaging. One simple way is we can measure the fecundity of males. Whether males are affected, females are affected. We can simply count in a given unit of time per fly how many eggs are laid, how many eggs are hatching out. And again, these experiments don't need, all you need is a way to grow flies and treat them through the food or by aerosol to some xenobiotic, and then see how it affects the fecundity of the fly. Again, simple experiments. Only thing is, it can't be done in a single day. It will need 2 to 3 days time to look at, give the treatment and examine the progeny and count the eggs and see how many eggs hatch, how many eggs die, so that one can make out. So it can be a very good assay for environmental toxicity or any genetic toxicity, any genetic factor. This can also be used to identify genes that control fecundity. If there's a mutation and that reduces fecundity, then we know that there's doing something with the spermatogenesis or oogenesis. Other way for immunity study, Drosophila doesn't have adaptive immunity like the antibodies that we have, or T cell or B cell, but they have the basic innate immunity. And that basic innate immunity is common in Drosophila as in humans. And in this case, there are a number of small molecules that act as defense against specific testing. For example, lipopolysaccharide is a common antigen that can be used in Drosophila, and it will elicit response of certain genes. For example, sarcopene is one of the innate immune response gene in Drosophila. And so if we have a transgenic line, sarcopene A, a promoter fused to LAG-Z of E. coli, and a transgenic fly. Now, if these larvae carrying this are exposed to, let's say, LPS or infecting bacteria, in the Malpighian tubules, which is one of the immune organs, and homologue of human kidney, but it also is an immune organ in Drosophila, an insect. And you can see synthesis of appearance of blue color, which indicate that the sarcopene gene has become active in response to infection, and so one can see the immune response being mounted. And what stage it mounts, when they are weak, when they are less. If you do the same thing at first instar, response is very weak, because first instar still is not ready with its immune response. But as it matures, the immune response becomes better. Then another interesting thing is the behavioral gene. Who many of us know as the person who did the autolocus fine structure gene, who gave the term cistron, recon Newton. Just before I go about Simeon Benzer, something more interesting. In 1940, Simeon Benzer was involved in making transistors. He was a physicist. In 1949, he turned into a fly geneticist. Straight from physics, he jumped to fly genetics because he heard a lecture of Sir Max Delbruck, and that made him convert himself into a biologist. And then he looked at the autolocus of T4 bacteriophage for about 10 years and came out with a brilliant explanation of recon, cistron, Newton, and so on. And after having done that, he decided to jump to flies. And then he asked this question. There's an article available of him that how he did it. he started looking at human behavior and saw that, okay, even his two daughters were twins, behaved in different ways. So his question arose that why should they be different? And he started using Drosophila for behavior. So he became the father of behavioral genetics and using Drosophila as the first source. And till date, Drosophila remains a very good model for neurogenetics, for behavioral genetics. And Obaid Siddiqui is sitting somewhere here. He learned, who set up the TFR fly lab, his behavioral genetics from Samuel Benzer when he visited him as a postdoc. And that's where the behavioral genetics started. And you see what is interesting is a fly can decide whether it should jump from this point to this point or should fly. It can do that estimation. And this is very interesting to think of. One simple way one can look at soft life without needing any fancy things is courtship behavior. So all that you need to put a mature female and a male in a petri dish and allow them to acclimatize. In 5 minutes, then you can see that the female is not already mated. It should be virgin female, but 4 or 5 days old virgin female. Put it with a male, and then within few minutes you can see that they start this stereotype behavior. Absolutely. But if there's a mutation, then something goes wrong. They don't behave like this. Or if the female is mated, it will not respond to the male's approaches. And so this becomes a very good way of looking at the behavior. For example, the lower panel showed the actual videographs, the photomicrographs of things happening inside a petri dish. You don't need any sophisticated equipment, just a small glass petri dish. One can look at how larvae move, whether their musculature is affected, because several diseases, neurodegeneration diseases, nerve diseases, or certain treatments affect their motor functions, affect their muscle contractility. So one can simply track, take images using even smartphones. One can take a small video and see how a larva has moved from here to here, here to here, or it remains moving on in a small area. And then one can do an image analysis using simple statistical tools to find out whether it's moving freely or it's moving in a restricted area. And this can give us an idea about the muscle contractility, the regulation of the muscle movements, and so any defect in muscles, any defect in neurotransmission, any defect in brain function can be directly assessed by this. And these are very interesting experiments for students to do in classroom. Similarly, we can look at that flies are typically negatively geotactic. That means they move away from center of gravity. So if you put flies in a tube, and wild-type flies will tend to move towards the upside. But if the motor functions are affected, for example, one can treat the flies with rotenone, which is one of the insecticide, and because it damages their neurotransmission, then the fly don't move up, they remain bottom. So all you need is simple test tubes or even a 10 ml pipette where you can mark a distance, all right, how much time the flies take from, you tap them down and then let them move, how fast they can come up to this. The ones which have compromised muscle movement, neurotransmission will not move up. And this can be a very good way to assess the behavioral part of it. One can look at attractant and repellent smells, because for insects, smells are very important. Olfactory sensation is very, very, very critical. And so all that one can do is one can put larvae in the center here, and on one side put a cotton dad with one volatile component versus another volatile component, and then see if larvae move towards one way or the other. If they don't move toward this side, that means this chemical is repellent. If they move more toward this side, this is an attractant. And so this is again a simple experiment which doesn't need any fancy equipment. All you need is just a small petri dish and some of these chemicals which can be used to look at the behavior. The Drosophila eye is a beautiful thing to look at. It's a compound eye with almost 700 eyes in each one. Now this is something interesting. We have each eye has one lens. Here you have each fly has two eyes, each eye with about 700 to 800 lenses. How does it coordinate its vision? It's another interesting thing. But the thing is that we can see this beautiful pattern. Earlier, people used to do this with scanning electron microscope. This image would be generated by scanning electron microscope. But an excellent discovery in our own lab by one of my students, Richa, who is now my colleague, she developed this nail polish imprint technique. All you need to do is take the adult fly, remove its head, cut its head. You don't need to do any fixation or anything. Just put a small drop of transparent nail polish on its head. Wait for a few minutes, and then using fine needles, crack open this nail polish which had dried up, and carefully remove the nail polish layer over one of the eyes. And go and see in the microscope, and you get a beautiful imprint of the H. ommatidium as it is there. You can see in higher magnification, and you see same resolution as one would get with scanning light microscope. And this simple technique, it needs just 5 minutes. All it needs is some flies, transparent nail polish, slide, and a cover slip. All you do is you take this nail polish layer, put on a clean slide, put a drop of 50% glycerol, mount a cover slip, go to microscope, bright field, or phase or DIC, and you can see these patterns, beautiful arrays. If there's any disruption anywhere, any gene that has gone wrong, or there's any neurodegeneration, or something that you treated the flies which affect the development, you can immediately see this disruption, as I'll show you. And so this model has been extensively used to study human neurodegeneration in Drosophila. We have models of Huntington's, Alzheimer's, Parkinson's, where the scar or the spinocerebellar ataxias in Drosophila. We have the mutant human genes put in Drosophila, and we can activate them when we want, using, again, a very interesting system of having one fly which expresses a protein called GAL4. This protein is from yeast. And this is put under a promoter called GMR. GMR promoter is expressing only developing eye disc. That's more very specific. And so this fly will make this GAL4 protein in its developing eyes. But this protein is a transcription factor. There's no target in Drosophila cells, and therefore it remains without any consequence. We have another transgenic lines in which we take the US upstream activated sequence again from yeast with which GAL4 binds. This is a transcription factor binding site. We take this sequence upstream of any gene that we want to express. For example, here I've written polyQ, that is a mutant gene that has a long stretch of glutamine residues, as happens in Huntington's, for example. So when we cross them, in this progeny fly, now this GMR will make the GAL4 protein made in this part of the eye disc, where the eye is developing, ommatidia are forming here. And in these cells, GAL4 will bind to the US sequence and cause expression of this polyQ protein. If this polyQ protein is toxic, you get degenerated eyes. If it is not toxic, you get normal eyes. So one can look at this. So any gene that affects eye development causes neurodegeneration, it becomes a simple SO. We don't need any molecular biological things. All you need is these flies, cross them, and look at this thing. And now, to look at this, you can use again the nail polish imprint technique. That if the arrays are normal or mutated, we can very clearly see them. And so it becomes a very simple way to look at if there is a neurodegeneration happening. So eye model has become the Drosophila eye has become a wonderful model across the world for looking at various diseases that affect neurodevelopment and differentiation. And this can be done again in classroom without any fancy things being required. So what I've tried to do here is given several examples. There are many more examples where Drosophila can be used, although it was initially used for studying in genetics, and that's what people still think that it's used only for genetic experiments. But now it's time that it can be expanded to many areas in biology. And to facilitate this, we are now have compiled this book, which will be an e-book freely available across to anyone from Indian Academy site. We hope that this will be available by end of this month on the web page. It's about 800 pages or so of e-book, no hard copy, and where each of these different practicals, or 85 classroom practicals, are described, which can be done. Some of them, of course, need a little more sophisticated facilities, but a large number we have thought should be which can be done by any laboratory which has some basic facilities and which can, and of course, as I said at the beginning, that there will be ways where these flies can be obtained by the designing colleges for doing their experiments, classroom. They need not maintain it over the years. If they can maintain, very good. But if they cannot, then the flies can be obtained from some of the stock centers or labs where they are being worked. And so these students can be exposed to them. So with that, I'll stop and take questions. I hope this has been useful in some ways. So I'm now ready to take any question that you want.
Thank you, Professor Lakhotia, for this excellent session. We have gone through the exciting journey of development of Drosophila genetics from the very beginning, from the first physical map to the developmental, behavioral, and neurogenetic aspects. Thank you once again. So, shall we take the question right now, sir?
Yes, I'll be happy to answer.
Okay. Participants, you can unmute yourself and ask questions to Professor. And in the meantime, let me read out the questions we have in the Google chat box. So, there is a question, sir, how to screen anti-diabetic activity in Drosophila?
Okay, that can also be done because there are models for diabetic studies that you can feed them on high glucose diet, create diabetic conditions, and then see what kind of treatments can reduce their thing. I'm not fully aware, but I know one person who is working in Lucknow, Dr. Naveen Gautam. He's using diabetic. There are some other people also who are using diabetic model in Drosophila, and this can be done. It has become a very good model now.
Thank you, sir. So the next question, how do we run Western blot analysis with Drosophila body?
Well, Western blot, you see the protocol remains the same thing. That you homogenize, take proteins, run them on gel, separate the proteins, and then transfer the proteins through as you do for either by simple diffusion or through electroblotting, and then use the specific antibody that you want to do. This ebook that we have will have a protocol on this also, a detailed protocol how to do Western blotting with proteins from Drosophila.
Okay. So which model of organism we should use and why?
Well, each model has its own advantage. Each model will have certain things that it offers, other models do not offer. And certain things that we discover in one model may or may not be applicable in other models or human beings if we are trying to look at it from human point of view. But then also it depends on where one is trained, what are easily growable, which can be maintained easily. And so that's what should be done. So I won't say that this model is better than other, but then the advantage of Drosophila is the numerous thousands of mutants that are available, transgenic that are available. And a remarkable thing that Drosophila community offers is with modern that all the mutants that are available are freely shared. Even newly discovered mutants, we all share with people without any IPR issues or patents issues or something. And there are stock centers which are there. Unfortunately, in India, we still don't have a typical stock center, but there are certain labs which will be willing to share the flies as you need it. And so, from that point of view, Drosophila remains a very good model.
The next question we have, is there any gene for aging in Drosophila, and what happens if we remove that or those genes?
Well, the point that we should understand, there's not one single gene that causes aging. Aging is a complex biological process, and there will be a large number of genes involved. And there have been a lot of research in aging, but then it's a kind of quantitative character, quite a variety of factors. And so we can't just remove one gene. We can have different genes because people are selected for lines that have long-lived flies. People are selected for lines that are short-lived lines, but they don't find a single gene mutation. It's a complex thing. But Drosophila is again a good model for aging studies because one can look at the change in brain, one can look at changes in metabolism, one can look at changes in the reproductive system, and so it has remained a good model. But I don't say that we can identify any single gene that this is the gene for aging. And for example, we ourselves show that some of the Ayurvedic formulation like made of amla can prolong life in flies also, as they have been claimed in humans. So this is one area where we can test for effect on the life quality as well. That Drosophila can be used for that.
Next question. What is transgenic fly?
Well, a transgene is actually a gene which has a DNA which is different from the host's own DNA. I mean, from that particular species DNA. So basically, we take DNA from some other organisms, manipulate it in vitro by genetic engineering as we wanted. We create a sequence and then introduce that into the host genome where we want to do it. And that becomes a transgene. And that organism is called transgenic. So if I say transgenic Drosophila, that means this Drosophila has its own genome plus something else that we have introduced in it deliberately by our choice. A gene taken from humans, a gene taken from another Drosophila species, a gene taken from bacteria or a plant or any other organism. That will be a transgene.
Okay, so next question we have, sir, how do we distinguish between male and female Drosophila? Is it depend on size, like the female one will be a bit large in size?
Well, male and female can be distinguished. First is size. Yes, females are larger than males, but then there are very distinct morphological differences. The posterior end of a female is more pointed, male is rounded. And male in Drosophila melanogaster, the tip of the abdomen is much darker, blackish in color, whereas in female it's not that color. There's no black color, no black melanin pigment there. Another important point between male and female is the presence of what is known as sex comb on the one of the dorsal segments of the first pair of legs, which a pair of stout bristles, which are present only in males but not in females. So that's the way we distinguish between male and female easily.
Okay, next question. Sir, what could possibly be the problem if fungal contamination, I think it's contamination, occurs for a transgenic line of Drosophila, but not when the same medium is used for native strain?
Well, I don't think that transgenic strains are more sensitive to fungal infection. No, that's not true. Because fungal infection not coming because of the transgene. Fungal infection is coming because there's something else is contaminating. So it's not that only transgenic lines will have fungal infection and the others will not have. They can have, and that's where fungal infection are rather difficult. We have a series of processes to be done that every generation we quickly change the flies to new vials and new bottles and keep on treating them with something. Actually, in the Drosophila food, we add nifedgin as an antifungal agent, which normally keeps fungus away. But if the fungal infection is strong, then we need to do some treatment. But it's not that transgenic lines only are sensitive to fungal infection. That's not true.
Okay. So, sir, how to count Drosophila neuron by flow cytometry?
Well, flow cytometry is can be used for anything, counting any number of cells. But the way neurons can be counted by flow cytometry, there are certain lines available in which neurons will be expressing GFP, green fluorescent protein, and they will be fluorescent. Or we can have different neurons only expressing GFP. So we can use such transgenic lines where we have GFP expression happening in either all neurons or a certain subset of neurons, and then we make a suspension of brain cells from a large number of lines, pass through the flow cytometer, select for GFP expressing lines. So these are the ways, but they are not absolutely 100% correct. There can be always contamination. And and you need a large number of flies to do. All the people have tried with fewer numbers, with much more sensitive flow cytometric system, and that is to some extent possible. But you need a marker, and that marker is provided in the transgenic system, whereby we express GFP or some other fluorescent marker in those specific cells.
The next question, so which method we can follow to isolate DNA from Drosophila?
For extraction of DNA, it's the same method.
Yes.
Homogenize with an STS containing solution, and then do ethanol, the phenol chloroform extraction and ethanol precipitation. The process is absolutely same as one would do for any plant material or any mammalian material or fish material or bacterial material. Only in plants, of course, you have the problem of removing cellulose in the first place. But in these cases, you don't need anything. All you need is SDS and then phenol chloroform extraction and finally precipitation with ethanol. Same method.
Okay. So what is the process to make a stock of CRISPR mutants in Drosophila?
Well, CRISPR mutants will require again a complex system. You need a variety of lies. Many of them are available now that they will express the Cas enzyme, CRISPR Cas enzyme. You need to create your transgenic with the guide RNA, and then cross them so that with the Galpo driver so that the CRISPR Cas becomes active in germ cells, and then it will do its own. So these standard lines are available. All you need to do is generate RNA and then make a transgenic that will express that particular guide RNA. And fortunately for this, now there are services available. For example, CCamp in Bangalore has a fly facility that can generate CRISPR-Cas lines for you on payment, which is not very cheap. It is a little expensive, but they can do it.
Okay. In human or animal, a cell dies after few division due to programmed cell death. But in case of amoeba, what it does happen, and they are called immortal.
Well, partly I think there's a big question, very very fundamental question, why amoeba doesn't die. But a part of the reason is that amoeba doesn't have typical sexual reproduction. The organ that introduced sexual reproduction introduce a limited life. Why biological system have to do that kind of system? But that is what we look in every organ. Many plants never die. You can have a cerebral propagation; they can continue. But the moment we come to sexual reproduction, there is a limited life, and that's partly because of the regulated form and function that we have. See, the prize that amoeba pages, amoeba doesn't have a face like us. We have a defined body shape. Amoeba doesn't have. But that's because it just keeps on dividing. Of course, one can say paramecium has a definite shape and it can also keep on dividing. But paramecium, after a certain number of bacterial reproduction, will not be able to maintain itself unless it undergoes through a conjugation system. So these are basic fundamental biological processes. But then what we know is that in all our system, telomere is one of the important points which defines how many times a cell can divide. So telomere maintenance becomes an additional issue that one has to take care in those who have finite life or cells that have finite division capacity.
So there are a few more questions. Shall we take?
I'm okay.
Okay.
If your time becomes...
No, it's not about our time. It's if you are okay, then we can proceed. So the next question, sir, what is the effect of amla in Drosophila and human gene or genes?
Well, what we looked at was amla ketosan, a very simple preparation, a standard Ayurvedic preparation. And that we saw that it does prolong life of flies. It makes them more heat shock resistant, oxidative stress resistant, as was expected. But more importantly, what we found was that it suppresses Huntington's symptoms in Drosophila models. Many polyQ models we check that, Huntington's or some of the spinocerebellar ataxias, and when these larvae were fed on amlodipine, where we were expressing these genes by our genetic program, and they were neurodegeneration, but when we fed them on this, neurodegeneration was substantially reduced. Not completely gone, but substantially reduced. And this we after doing this, we also looked in rats in collaboration with CCMB, and there too it does similar thing. So amla is certainly, I would say, a good medicine for brain and for life in general, as long as it is prepared in a careful manner. When I'm mentioning this, I should also caution people that many of the commercial products that come in name of Chyawanprash, different so on and so forth, are not properly prepared and can actually be harmful than beneficial. But a well-prepared things of amla will be very good. One can take even raw amla without having all these fancy preparations. That cannot be equally good. And that's what we learned from Drosophila again. We provided the evidence that yes, it does. And the mechanism. We could look at what kind of genes are activated, what kind of genes are not activated, and why should it provide that advantage. These we have published already.
Okay, so next is how Drosophila study contributes in cancer genetics.
In translational genetics.
Cancer genetics.
Cancer genetics. Well, we have models of cancer. Like many labs, a large number of labs are working on cancer using Drosophila model. The advantage in Drosophila again is we can induce cancer when we want. See, one can induce cancer in Drosophila also by treating with something, but then there is a more important is that you can genetically program that at this particular moment, a cancer cell will be generated. There are genetic tricks that we can do that. And that's what we are also utilizing, that we create clones of cancer cells, and then we can follow their progression. We can follow what kind of treatments will reduce them, what kind of things that can happen. So Drosophila has become a great model. And in fact, historically, it was only studies in Drosophila that gave the concept of oncogenes and the oncogenic suppressor genes, tumor suppressor genes. It's because of studies in Drosophila that this concept started. And so Drosophila remains a very good model. And as we learn more about it, as we develop more kinds of transgenic lines and other things, it becomes more and more attractive.
Okay, next is, sir, is there any method for the cryopreservation of Drosophila embryos with promising viability?
No, so far, cryogenic preservation has not succeeded. There were major projects taken up in the last century, but none of them succeeded. That's one limitation, that we cannot cryopreserve flies or embryos. So far, no. There's no technique available.
Okay. So the last one, can we conduct any medicinal tests in Drosophila which we conduct in human? Will be the results are same or not?
Well, what kind of medicinal tests? I mean, you see, if you are doing some biochemical tests like estimating sugar, estimating fat, and then the techniques remain same, no matter whether it's the sample comes from human or from this thing. But to diagnose a human, we need a human to be tested. But then the studies can be done. We can simulate parallel studies in the Drosophila flies, and then gain information, and then make their use with certain clear understanding in humans as well. That's where the fly models become important. That we have fly models for several heart diseases, we have fly model for obesity, fly model for diabetes, fly model for a large variety of neural disorders, muscle disorders, and so on and so forth. The models are available. So one can do that kind of study in them and then see how much of it can be applicable to humans.
Okay. Is there any other question? Participants, you can please unmute yourself to ask question. I think there is no more question. Salma, is there any question in YouTube live?
So there is no questions.
Okay. So let me thank our honorable speaker for this excellent session once again. Thank you, Professor Lakhotia.
I'm really happy that you people are doing this kind of activity. Keep doing well. And anybody can feel free in writing to me anything that we can help. It will be our pleasure. Do well.
Okay, that is great. Thank you, sir. Thank you once again.
Thanks, sir.
So we are concluding the session. And dear participants, please follow the WhatsApp group for tomorrow's lecture and all other details.
Okay,
So thank you everyone.
Bye.
Bye bye.